Hello everybody, welcome to Knowledge Without Bar. After a long time, I came up again with a completely new video, and today we'll discuss about enzyme immobilization. It is very important as well as very interesting topic. So before I go into further detail, please subscribe the channel. So what does the enzyme immobilization means? As the term implies, enzyme immobilization means uh, something that is immobilization of the enzyme. We are going to stabilize the enzyme. Now, what is the requirement of this enzyme immobilization? We all know that enzymes are very important. It has several different applications, starting from the industrially produce several products. Not only that, enzymes, free enzymes are also have some pharmaceutical activities. We are taking many enzymes as a medicine, like gastric enzymes, etc. But one problem is enzymes, these free enzymes are not very stable because enzymes are highly reactive and they have a certain half-life. So this is a real problem in industrial applications of the enzyme to store the enzyme for a long time. So enzyme immobilization is nothing but um, a process of long-term storage of the active enzymes without compromising its activity. So what is enzyme immobilization then? If I make it simple, enzymes are used in different industrial productions, but the isolated enzymes, that is free enzymes, are very poor in their stability and activity. Hence, to overcome this problem, free enzymes are inserted into the semi-solid carrier before using them in the industries. This process is called enzyme immobilization. Being more technical, the definition will be the imprisonment of an enzyme in a distinct phase. Now, distinct phase means in a particular phase of the enzyme production when the enzyme is capable of showing its full activity. Because every enzymes, we all know, they are the product of some biochemical pathways. So they have some precursors, and these precursors are uh, again produce some other molecules. So distinct phase means in that phase of the enzyme when you're going to entrap that or immobilize that enzyme when it has its full potential to break down the substrate. Now, in a distinct phase that allows exchange with but is separated from the bulk phase in which the substrate effectors and inhibitor molecules are dispersed and monitored, is called enzyme immobilization. This definition is somehow it's difficult to understand, but you can use this definition. It's very simple and straightforward. So simply, the motive of the enzyme immobilization is increase its convenience, its economy, and its stability. That is important. Now, the first commercial application of the enzyme immobilization was reported in 1969 in Japan with the use of the amino acylase. This enzyme is highly used in Japan, and this enzyme was produced from and is produced from Aspergillus oryzae. Because its uh, excessive commercial use, starting from the shake, miso, soya sauce, that is the fermentation technology, fermentation industry, enzyme production, due to the huge application of the Aspergillus oryzae in Japan, in 2006, this Aspergillus oryzae as considered or um, designated as Japanese national organism. It was uh, approved by the Japanese Brewing Society of the Japan on October 2006. So this is the first time in 1969, uh, the first time enzyme was immobilized uh, from the Aspergillus oryzae, and the enzyme was amino acylase. Now the process of enzyme immobilization. This is I'm there are several things we can discuss within this topic, but I make this video very straightforward, uh, which will be required for our examinations and understanding of the basic enzyme immobilization. So the process of the enzyme immobilization is very important, and there are two uh, major process. We can distinguish it in physical method and chemical method. And within the physical method, the enzymes can be adsorbed enzyme or entrapped enzyme. And the chemical method, it can be covalently linked enzymes or cross-linked. We're going to discuss this process elaborately, but this is the major uh, classification of the process of enzyme immobilization. So within the physical is adsorbed. Adsorbed means, as the term implies, it is on the surface of the substratum, and entrapped means it is engulfed within the substratum, but it not react with the substratum. So the important point is the substratum should be non-reactive in all these physical processes. And the chemically, it's in the covalently linked. Again, the enzymes are on the surface, and it forms a covalent linking or covalent bond with the substratum. And cross-linked means uh, the chemical molecules which bind with the enzyme molecules, it also forms the linkage either with the cross-linked molecules or with the enzymes. Sorry. Now the forms of the immobilized enzyme. Uh, it means uh, what are the forms after the enzymes are immobilized. There are four major forms. It may be after immobilization, it may be in capsular form, it may be in form of beads, it may be in form of the column. This column enzyme, immobilized enzymes in column are popularly used in laboratories for isolation of different proteins, different enzymes, which is called the column purification. And another is the enzyme in membrane. This is also um, popularly known as dialysis. Uh, in in laboratory, we are very frequently use this technique, the dialysis technique, to uh, purify the isolated enzymes. We put the isolated enzymes into a semi conductive membrane, and we pour it or dipped into a buffer system. So the due to the uh, osmosis, the enzymes are remain inside the this semi conductive membrane, and these buffer systems are interchanged within the membrane system, and ultimately after some time we can get the pure form of the enzyme, because all the impurities can come out from the membrane. Now this is the example of the physical method. As already told, it may be adsorbed or it may be entrapped. Now for the adsorption, as I already said, the substratum should be solid. So a solid inert polymer as immobilization surface was used. For example, maximum cases we use the cellulose based materials, ion exchange resins, porous glass, silica gel, charcoal, etc. Onto the surface, the enzymes are simply attached with a very weak bonding pattern. And in the entrapment, the substratum actually engulfs the enzyme material, but without modifying its properties. It it forms a very weak interaction patterns. Uh, the interaction may be electrostatic interaction, hydrophobic interaction, hydrogen bonding, without hampering its enzyme's property. So uh, it may be entrapment, that is, it forms when the substratum forms electrostatic interaction or hydrogen bonding, etc. Or it may be a semi-permeable membrane, as I already said, the semiconductive or semi-permeable membrane system are used in the enzyme membrane. So we can use a membrane, the semi-permeable membrane, and the synthetic semi-permeable membrane, and we engulf the enzymes within this membrane. So this process is without any interaction. Any of these interaction are usually not happened in case of the encapsulation. This is simply uh, just separate the enzyme molecules from the outside environments. It's nothing but. So this is the physical method. So it may be adsorbed or it may be entrapped. Entrapped means either it completely interacting with some electrostatic, hydrophobic or hydrogen bonding interaction, or it's simply encapsulation method. So uh, this is the example of this is encapsulation process, which is liposomal entrapment. As in case of the entrap or encapsulation or entrapment, we use the synthetic membrane. So the synthetic membrane, in case of particularly the drug delivery system, where if we use the synthetic membrane, it can cause some side effects within the human bodies. So scientists are always thinking about some entrapment system, in particularly the drug delivery system, uh, in particularly used in some drug delivery system which have minimum or no um, side effects with the human body. So the best entrapment system is liposome. Liposome is nothing but a artificial membrane. It can be originated or developed from cholesterol or natural non-toxic phospholipids. So as you all know that phospholipids bilayer have two uh, tails. One is hydrophilic tails, which uh, protruding outside in two sides, and the hydrophobic tail, which are um, embedded inside. So this artificial membrane originated from the lipid molecules or cholesterol or natural non-toxic phospholipids are called the liposomes, and we can entrap some hydrophilic enzymes within this liposomal system, and also we can uh, entrap the lipophilic or hydrophobic enzymes inside the same liposome. So this the utility of the or the benefit of this liposome is it can deliver both hydrophilic and lipophilic or hydrophobic enzymes within the same delivery system. Another thing is that as it is originated from natural lipid molecules, so it has minimum or no side effects with the body. So this is a typical example of the enzyme entrapment. We can discuss more about liposome. It's a very interesting phenomenon, a very interesting topic. We maybe discuss later on regarding this topic. So now come into the chemical method of enzyme immobilization. The first point is covalent bonding. If I go back, so chemical uh, immobilization may be of two types, covalent and cross-linked. So this is covalent bonding. So you all know covalent bonding is uh, the enzyme immobilization by covalent bonding means we are using some substratum which are covalently linked or covalently interacting with the enzyme, and this covalent bonding uh, maybe this chemical method of enzyme immobilization are classified by four different types. One is diazotation, that is bonding with amine group like amine benzyl cellulose amino derivative etc. It may be of peptide bonding, that is carboxyl group and the amino group linkage. Number 3 is group activation, that is cyanogen bromide is used to support the glycol group of the enzyme, or it may be polyfunctional agent, for example glutaraldehyde, etc. This is a covalent bonding, but it's also called the cross-linking phenomenon. We are going to discuss in the next slide. So typical example is sometimes a porous glass. Uh, Glass is, you all know that glass is inert substance. The glass may be activated with a sulfonate group. So sulfonate group have a negatively charged moieties. So if we're going to immobilize a negatively charged protein with it, so we just attached a amino group with this negatively charged enzyme molecules. For example, D-amino acid oxidase is fused with positively charged molecules. Okay. Now these positively charged molecules are easily interacting with this sulfonate group, and this enzyme can be immobilized onto the porous glass, and which is completely inert, and we can store this enzyme for a long time by using this type of method. Now is the copolymerization, which is also called the cross-linking, that is utilization of the polyfunctional agent. So copolymerization means we can use a chemical substance which can interact with the enzyme along with uh, other copolymerizing molecules. For example, this is suppose a free enzyme. We can use a copolymer or polyfunctional group, for example genipine. The uh, properties of genipine is it can interact with the free enzyme, so it can bind with the free enzymes and also it can bind with themselves, so forms a copolymerized enzyme group. So by this phenomenon the entire free or soluble free enzymes can be copolymerized. Another method is, for example, this is a free enzyme and this is glutaraldehyde. Glutaraldehyde also used as a copolymer substance or multifunctional group. So glutaraldehyde on the other hand, it interact only with the enzyme molecules but not cross-linked with each other. So this is also copolymerized enzyme. So these two type of copolymerization or cross-linking can be performed in case of the immobilization of the enzyme. So at a glance, the enzymes can be immobilized onto the surface. So it may be by adsorption, which is a physical method, or maybe by covalent bonding, as we discussed, is a chemical method, or maybe entrapment with uh, synthesized substances, or microencapsulation or use of the liposomes by natural lipid molecules, or we can also go for the cross-linking. Now this is also very important. What is the features of the enzyme immobilization or immobilized enzymes? First of all, the immobilized enzymes are more stable than the free enzymes or soluble enzymes. Number two, they are more stable towards heat, urea denaturation, ethanol, pH activity or pH variability. They produce more potential reactions than the free enzymes. They are usually separated along the carrier during harvest of products, and the loss of enzyme is very much reduced. These enzymes are easily released from the products into the medium by simple diffusion. Number 5 is these enzymes need no modification during the catalytic reaction. And number 6 and most important is that these enzymes obey strictly the kinetic characters of the enzyme, that is explained by the Michelson-Menten equation. Now the application. There are several application of immobilization of the enzyme. one paper came in 2018 which describes a very beautiful paper. Uh, the immobilization technique is uh, very much is used in the future medicine as a flexible sensor array development of sensor array development of like detection of the alcoholic person, uh, now electrochemical detection of the ionophoresis, which is called, and another is, for example, this is a there are lots of enzymes are immobilized onto the surface. So simply if we drop a single drop of blood in this detection system, this will diffuse inside the substratum, and in each of this block there is separate enzymes are immobilized, and if uh, the particular target molecule or target substrate present within the blood sample, it will interact or react with the enzyme immobilized within the block and give the result. So simply using the enzyme immobilization technique, a single drop of blood can give you the result of different or arrays of the enzyme reaction. This technique is hugely used in detecting the narcotics. Again, it has also have application in the food industry, dairy industry, pharmaceutical industry, and enzyme therapy. Thank you for hearing. Thank you.